and one of the function is they function as adapter molecules. So why are they called adapter molecules? Because they link, one transfer RNA links itself to one amino acid and it transports the amino acid to the ribosome, where the ribosome, on the ribosome, a peptide bond is formed between the growing chain of the protein, and this transport of amino acids is done by transfer RNAs. So each type of transfer RNA attaches to single amino acid. What it means is that one RNA, one type of RNA or one kind of transfer RNA can link only to one amino acid. So the same RNA cannot bind a different amino acid in the next cycle where it's still supporting protein synthesis. You get that? So one transfer RNA can only bind one amino acid. The same RNA can bind the same amino acid a second time, but not a different amino acid a second time. Okay? Such RNAs that accept the same amino acids but have different anticodons. Okay. All right. So here I want to speak about. Maybe I should put that anticodon. Okay. Look at this, guys. So if you look at it, this is the one in green is your messenger RNA, right? And this messenger RNA has the codon UCC and so on, so on, so it has UCA. Now this is referred to as the codon on the messenger RNA. At the very base of the transfer RNA, at the very tip of it, it has nucleotides which are complementary to the codon. So if you have UCC, you have AGG here. These three nucleotides are referred to as the anticodons. So the base of the messenger RNA, sorry for jumping between slides, but okay, look at this. This is the transfer RNA, right? The very base of the transfer RNA has anticodons. So these nucleotides are complementary to the nucleotides found on your messenger RNA. Okay? Sorry for moving back and forth. Okay. So transfer RNAs that accept the same amino acid but have a different anticodon. Why do they have a different anticodon? Because in the previous slide I said the genetic code is degenerate. More than one codon codes for the same amino acid. If more than one codon here is coding for the same amino acid, so if you have 4 different codons here, right? Then you should have 4 different anticodons, right? But, so such RNAs, such transfer RNAs that accept the same amino acid but have different anticodons are referred to as iso-accepting transfer RNAs. Is that clear? Iso-accepting transfer RNAs. Now, here is an example of showing you a codon and an anticodon and how that UCC codes for serine. Is that true? UCC. Let's go back. UCC. Where is UCC? Where is UCC? UCC. UCC codes for serine. Yes, that's true. UCC codes for serine, and usually the binding between the third codon and anticodon is actually not that strong, and for some reason. That's why it's referred to as it's called wobbling, or it's called the wobble position. Third nucleotide is not strongly bound. Anyway, now finding from early studies of the genetic code indicated that the genetic code is non-overlapping. Generally, I worded the word generally non-overlapping because if you look at this here, these three, this is one codon, 2, you have 3 codons here, 1, 2, and 3. So each codon codes for one amino acid. Okay? So here is AUA, CGA. But actually, if you skip the A, then you have UAC, which is actually an entirely different codon compared to the first one. This is called overlapping. Usually in humans, the concept of non-overlapping does not exist, at least in eukaryotes. So basically, if we have 9 nucleotides, we will see this as 3 codons, and each codon will give one amino acid. That's clear. But here in humans, we will not skip, the human RNA does not skip A, and it does not start reading from UAC and give a different amino acids like shown here, tyrosine. But this kind of overlapping concept is actually seen in virus, because the viral genome is very small, so the virus has to find a way where it can use the 9, you can use this 369 nucleotides, and instead of just creating 3 amino acids from the 9 nucleotides, it finds a way by moving, by reading the second set of 3 nucleotides and giving rise to different amino acids. Is that clear, guys? So the first codon is skipped. If the first nucleotide is skipped here, you can create 3 different proteins. If you skip the first and second nucleotide here, you can create 3 different amino acids. You get that? So it's basically kind of a frame shift mutation, where you kind of, the frame is entirely shifted by moving one. So such overlapping mode of the genetic code is usually seen in virus, but in eukaryotes, we don't have that overlapping code, okay? The overlapping mode of translation. An overlapping code would be one in which a single nucleotide might be included in more than one codon as follows. Each different way of reading the sequence is called a reading. Each different way of reading the sequence is called the reading frame. So this is reading frame 1. This reading frame 1 has these 3 amino acids. If you skip the A, then this, and the further will actually become the second frame and the third frame, so on and so forth. So there are potentially 3 different reading frames in a given messenger RNA, but usually eukaryotes do not follow that mode. So the first codon is referred as the initiation codon, or called the start codon. The initiation codon, start codon, the first codon. Usually it's always AUG. Usually always. AUG is for methionine. Okay? Very rarely you have GUG and UUG, which is valunar glucine, but almost always the first codon is methionine. So if I ask a question like in multiple choice, something like AUG is always the first, methionine is always the first amino acid in a given protein among all species, then it's wrong. Okay, if I say AUG is the first amino acid in eukaryotes or in humans, something like that, it's right. You get it? Guys, is that clear? So, but if I ask the question, I'll be clear anyway. So termination codons are UAA, UAG, and UGA. They do not code for any amino acids, so there is no transfer RNA with anti-codons for UAA, UGA, UAG, and UGA, and this actually specifies the end of translation itself. So there are no tRNAs which have anti-codons to create with termination codons. Here is a table column showing you the codons. For example, humans, UGA is the stop codon, but the same UGA can code for tryptophan in some species. This is called alter code. So basically, see the bottom line here. The universality of the code near universal with some exceptions. The genetic code shown here is almost universal. Basically, the same codons will code for the same amino acid with some exception which seen in some species. Is that okay, guys? Yes, you know what? We can finish the remaining part of the chapter in the next lecture. It's okay. It's only 8, 10 more minutes. I know it's too long. It's okay.